The title of my presentation today is Mechanism and Oncogenic Role of Lysine Demethylase KDM5B in Prostate Cancer. Prostate cancer is disproportionately striking more on African American men than any other ethnic groups. As you can see from this slide, African American males die more on cancers than any other populations. And more importantly, on the left of the slide, it shows prostate cancer rate, the death rate is higher in African American population. As shown this blue, is almost 2 times compared to Caucasian population. During last 20 years, this trend has been like this, not very much improved. So there is certain reasons we would like to understand what's caused the disproportional and disparity on African American population. My lab works on cancer genetics and epigenetics. KDM5B is one of them. We work on this. And KDM5B is amplified and mutated in many human cancers, including prostate cancer. As you can see from this slide, the red color indicates amplification of KDM5B genes, and the green indicates the mutation of KDM5B gene. And the first one you can see is NEPC, which is neuroendocrine prostate cancer. Anyway, the amplification of KDM5B is high, and we would like to know, is this the driver of the cancers, or is standby markers? My lab moves forward one more step to detect the protein expression of KDM5B in prostate cancer specimen, including African American men and Caucasian, by collaborating with other investigators from other schools. As you can see from this slide, the KDM5B levels are significantly increased in cancers, in high-grade prostate cancer, as well as the metastasis samples, which is consistent with the CBio portal data shows KDM5B is amplified in prostate cancer. What is KDM5B? KDM5B, also called JL1B or PLU1, is a demethylase and remove methyl group from histone 3 lysine 4 trimethylation or dimethylate to histone 3 lysine 4 monomethylation. It contains several domains, as you can see from this slide, GM, JN, AR, ID, and PhD, such on. So it's differently expressed in different organs in human samples, and so it shows differential functions in different tissues. We study prostate cancer using mouse models. P10 knockout mice have been well accepted and is showing 100% penetration in all prostate. So it's a good mouse model to study the cancer progression, disease progression, and so targeting. If there is additional loss of tumor suppressor on the top of beta-NOS, there is acceleration on the tumor progression. If there is a tumor suppressor or oncogene that can block the cancer progression, then we see the decreased cancer progression. As you can see from this slide, the wild type mouse prostate gland shows a slice single layer epithelial cell, and the prostate cells proliferate, and the prostate gland is filled with proliferated epithelial cell. The interesting is KDM5B and P10 double mutant mice showed the decreased phenotype. A few cells and the glands show the proliferation we call high grade P maximum or hyperplasia. At the same time, at the same age of the P10 mice showed aggressive prostate cancer. To understand what's going on in the decreased phenotype of P10 and KDM5B double mutant mice, we move forward to detect AKT. Phosphorylation of AKT has been recognized as the gold standard upon P10 loss in many organs, including prostate cancer. So as you can see, the phospho-AKT levels are lower in the P10 and KDM5B double mutant mice. And at the same time, the P10 mutant mice showed 100% all cells and the lyse location of phospho-AKT, and the wild type and KDM5B, they don't show any elevation of phospho-AKT. We perform the Western blot to understand what's going on and to know the molecular cascades upon the KDM5B knockout in P10 mice. So we got P10 and the KDM5B double mutant. You can see here the Western blot shows KDM5B levels are elevated in P10 now mice, and as well as phospho-AKT. And KDM5B knockout, we see the decrease of the phospho-AKT, and more important than the P10 and KDM5B double mutant show the decrease phospho-AKT and PI3 kinase, P100 alpha and P85. These two proteins are considered KDM5B impact factors, which are PI3 kinase complex. So this slide shows the quantification of the reduction of PI3 kinase. And we try to understand why this KDM5B is so important for PI3 kinase and AKT signaling. We use human prostate cancer cells, to take advantage of the CRISPR-Cas9 technology to generate KDM5B knockout cells. As you can see from this slide, the KDM5B, the Western blot shows the KDM5B band is not there. That indicates KDM5B knockout is there. Is not there. KDM5B is not functioning. And we perform the sequence analysis to ensure the gene, the target part of the KDM5B is deleted. So we are pretty sure KDM5B is knockout. And similarly, the most important for us is PI3 kinase and P100 alpha and P85 also decreased upon KDM5B knockout. This is the quantification. And KDM5B knockout decreased cell proliferation and effect the decrease of the PIP3 levels, which is the factor affected by PI3 kinase. So when the PI3 kinase is not functioning or reduced, the PIP3 levels are decreased. We perform a signal analysis to show what's the effect of KDM5B knockout on the cascades or the downstream target. We show the RS1, and indeed the peaks of PI3 kinase are affected. And ChIP-seq results show PI3 kinase directly affect the promoter region of PI3 kinase gene, which is called PIK3CA, which encodes the P100 alpha protein. And that effect is not caused by histone 3 lysine 4 trimethylation because KDM5B is a demethylation. KDM5B knockout will increase the histone 3 lysine 4 trimethylation. Most important, what does that mean for this genetic study? In a KDM5B knockout decreases cell proliferation, decrease the cancer progression of P10 mice. So in this case, the inhibitor of KDM5B can be meaningful for us to stop or block or reduce cancer growth. So we collaborate with our collaborators from other school and then use the KDM5B inhibitor to suppress the prostate cancer cells. As you can see from this slide, the proliferation of CYP2B cells, non-CAP cell, PC3 cells are induced by the KDM5B inhibition. So we use the P10 mice. As we know, the P10 mice showed the increased levels of KDM5B. So we inhibit the tumors by the KDM5B inhibitor. We show the tumor growth is significantly reduced by KDM5B inhibitor. And this also shows the KDM5B affects the PI3 kinase ubiquitination. And FOXA1 is also affected by KDM5B. So overall, of this study, the take-home message is KDM5B is important for prostate cancer progression. KDM5B is disproportionately elevated in African American prostate cancer specimen more than other ethnic groups. KDM5B is important for PI3 and AKT pathway. Inhibition of KDM5B suppress the tumor growth and the cell proliferation. I also would like to thank people in my lab and RCMI leadership and my collaborators from other schools. Thank you.